All right, ladies, so we're going to do some notes on meiosis, which connects with chapter 10 of your textbook. I apologize for the Christmas theme. We're a little early here, but we actually used to get to this a little bit later, and it was always after Thanksgiving. And so a couple of years ago when I made these slides, I made them Christmassy, and one of my rules is that once something is Christmas, you never go back. So I'm not about to make this into a Thanksgiving PowerPoint or something like that. So the goal here is to hit the highlights quickly of meiosis. I'm confident that you guys can kind of take care of that yourself for the most part, and then the main focus of these notes will be on a really important piece of meiosis, which is the introduction of genetic variation. So this is actually my old house when we used to do a competition in our neighborhood for decorating our houses. I'm a very classic traditional guy, like all white lights. And actually on my house now, I don't have a good picture, but I think I actually put more lights on my current house, but it's a bigger property, and so it doesn't look as impressive, I don't think. So I've just continued to leave this up here. But meiosis is the way that our body can create cells that we need for sexual reproduction. And ultimately, what we're going for there is cells that only have half of our genetic complement. So let's take a look at what we need to know for AP. This first slide is just some sort of vocabulary reminders. I'm confident that you guys will know some of these to begin with, but just to make sure, I wanted to compile them from chapter 10. Remember, genes are your pieces, your segments of DNA. Probably a newer term for you here is this idea of a locus. Locus basically just means a location on a chromosome. So a gene will occupy a certain locus, and we'll use that word a lot, honestly, until probably March. So you do want to make sure that you know that and understand what we're talking about with this vocab term locus. Gametes are sperm and eggs, right? So gametes are sex cells. And what's really unique about a gamete is that it only has half of the genetic complement of the organism, because these are ultimately going to come together to form a new organism. And so, as you might recall from freshman year and things, you are a combination, half your chromosomes from mom and half from dad. In order for that to be a reality, we need to make cells that carry only half of our chromosomes. Now, somatic cells is the term we use for the sort of like our normal body cells is a good way to look at it. Somatic cells in humans will have 46 chromosomes. So these are things like skin cells and fat cells and brain cells and bone cells and whatever. They have 46 chromosomes. But gametes, that means, are going to carry only 23 chromosomes for humans. And so we need a process to create a cell that only has 23 chromosomes versus 46. And that's where meiosis comes in. A couple other terms. A karyotype is basically a display or an image of all of the chromosomes in pairs. Those of you who did biomed, you looked at karyotypes some in there. Freshman year, we look at them briefly, but this is just a good way to visualize the chromosomes, and we'll see these again kind of throughout our next couple of units. Now, from this, it's very easy to see homologous chromosomes. And homologous chromosomes are each pair of very similar chromosomes. Essentially, if you have a homologous pair, one of those came from your mom and one of those came from your dad. And the way that we visually pair them up are these three things here. They're going to be roughly the same length. They're going to have roughly the same centromere position, which means a little bit of sort of a pinch here that you might recall from previous years. And then they're going to have the same banding pattern or staining pattern. When they get chromosomes, they will stain them, and what you'll see is that if they're homologous, they're going to have roughly the same pattern of stain. Sort of like different species of snake have the same pattern of stripes on them. If they're homologous chromosomes, they're going to have a similar pattern of stripes. Now, more vocab terms we have. Don't forget autosomes versus sex chromosomes. So you carry 22 different autosomes in pairs, or 22 different pairs of autosomes. Those are the ones we number. So chromosome 1, chromosome 2, chromosome 3, that sort of thing. And then you have one pair of sex chromosomes. And for females, that would be XX, and for males, that would be XY. And we'll get into that more after Thanksgiving break. Last vocab terms to know is diploid, which means chromosomes are all in pairs. So that's 2 times some number, versus haploid, which means chromosomes are not in pairs, but only a single set. So again, when we look at our cells, all of our somatic cells are diploid, and our gametes, the goal is that they should be haploid, or only carry half the DNA. So here's some pictures to go along with the last slide. I also compiled a little sort of like additional meiosis illustrations document that I posted on Schoology that you can open up. But these all come from your textbook too. So when we look at a pair of homologous chromosomes here, like we said, they're going to be very similar. They're going to have a similar centromere position. They're going to have a similar banding pattern, and they're going to be similar in length. And then this is a picture of a karyotype. This is actually a special kind. You don't need this term, but this is called a spectral karyotype, where they put all these different rainbow colors on it, basically. But it makes it even easier to tell which ones are homologous. And so again, you would have your 22 numbered pairs of autosomes, and then you'd have your single pair of sex chromosomes down here in the right-hand corner. Now, also, when we bring up this concept of reproduction, we have to take a moment to look at life cycles, because different types of organisms on our planet have different life cycles. And this basically looks at what we call reproductive history of organisms. Now, in a sexual lifestyle, a sexual life cycle like humans have, you alternate between fertilization and meiosis. But really, too, that's what we say, but you think of it like meiosis comes first, right? So as an organism, you can create gametes, and then fertilization is those gametes coming together. All right, now, there are 3 types of sexual life cycles in nature. There's the sort of animal or mammal kind, which is what humans would do. There's the plant and algae approach, and then there's the fungal approach. And again, all of these are sexual, meaning that offspring come from the joining of 2 cells, but they all work in a different way. So in this picture here, you've got this human or animal life cycle. Notice that the adult organism here can do meiosis in the reproductive organs to generate gametes, whether those are sperm or eggs. Then you have the fertilization stage, where those 2 cells come together to form this new diploid organism. And the new diploid organism develops to reproductive age, and you can start the cycle over again. So the human life cycle obviously is one that you're a little more familiar with from previous classes and things. Now, if we compare these 3, though, animals versus plants versus fungi, animals are what we just mentioned in that picture. You have cells called germ cells found in your reproductive organ that can undergo meiosis and create haploid gametes. These haploid gametes then come together in fertilization to form a diploid zygote. And then that zygote undergoes mitosis to create this diploid multicellular organism that is us. Now, here's how the other ones differ, and you don't have to know a lot of detail, but it's kind of weird. Plants, right? Plants do something called alternation of generations. I would recognize that term. It's very important now, and it'll actually be pretty important later this year when we get into some more plant science. Alternation of generations means the plant alternates between a diploid and a haploid multicellular stage. So again, for animals, you got this, diploid organism that can make a single cell that is haploid used in reproduction. But things like plants, right, can have a diploid multicellular stage and a haploid multicellular stage, and they alternate between the two. The diploid version called a sporophyte and the haploid version called a gametophyte. And again, we'll get into this more in the second semester, but two separate multicellular types of organism that is the same thing, really. And then fungi and protists are sort of the opposite of us, right? Their only diploid stage is a single-celled zygote, and then their haploid stage is multicellular. So essentially, you've probably heard of spores before. That's what's going on in a fungus, is they've got this multicellular haploid that forms a single-celled diploid thing, and then you see multicellular haploids again. So another picture that will show up on that extra illustrations handout, but just another way of viewing what we saw on the last page. So we got the animal life cycle here, which is just like what we do. You've got this plant life cycle that you notice is kind of cut right down the middle, where there's a diploid stage that's multicellular, and there's a haploid stage that's multicellular. And then the fungal one is almost the opposite of the animal one. So you got this single-cell diploid stage, and then you've got this more complex haploid stage. But I can tell you, too, you need to know very little about C. But definitely for now, we're focused on the animal life cycle, and later this year, we'll get more into the plant life cycle, too. All right, so meiosis. As I said, I'm going to kind of leave this up to you for the most part, because you might remember from freshman year, it's super similar to mitosis. But the point of meiosis is to try to cut the number of chromosomes in half, so go from diploid to haploid. And in order to do that, we're going to need two division stages. If you think back to mitosis a little bit, so I'm going to try to fit this up here in the corner. In mitosis, you started out with a cell that had 46 chromosomes. Then during interphase, you duplicated that, right? You had that S stage, and we went up to, at least for a short period of time, 92 chromosomes. Then mitosis divided that into two cells with 46 chromosomes again. So what we said when we talked about mitosis is that it was a cellular division that provided offspring that were identical to each other and their parent cell. Well, meiosis is going to start pretty much in the same way. Right? We start with a cell that has 46. It also undergoes interphase, so S stage and get up to 92. But now, remember, our goal in meiosis is to create cells that have half of the genetic complement. So in order to do that, we're going to have to, so this is meiosis, we're going to have to do two division steps, 1, 2, and we're going to finish up with 4 cells. And so this picture here, which I know I've now written over is just a great simple illustration that shows you this. Here's interphase with duplication. Then meiosis 1 is going to split the cells once. Meiosis 2 is going to split those cells again. And at the end of the day, what AP is most interested in is not a super specific step-by-step understanding and knowledge, but more of just Do you understand the difference between mitosis and meiosis? And do you understand the difference in the outcome of mitosis and meiosis? So I'll let you kind of look through this stuff, but so we'll skim through this, but I'll let you read these slides. There are some differences in meiosis 1. Synapsis means that we pair up the homologous chromosomes, and during synapsis, you get this really important thing happening, which is crossing over, or genetic recombination, where there's trading of pieces of the chromosomes between these different copies. When you get crossing over, you get something called a chiasma. So if we have kind of like one chromosome and a second chromosome, this crossing point is what's called a chiasma. Metaphase 1, really similar to mitosis, still the midline. But remember that since we're doing meiosis here, our midline is going to be 2 by 2 homologous pairings of chromosomes. And then at anaphase 1, instead of splitting in X, you're actually just going to send one of those homologous duplicated X's each direction. Telophase and cytokinesis, we split into 2 cells. Realize that at this point, we've got 2 daughter cells that are haploid, technically, but we just still have duplicated DNA. All right, so we need to break those in half before we really have the right count of chromosomes. And then meiosis 2 is really just like mitosis. Each of the daughters from meiosis 1 is now going to divide again, but it looks a lot like mitosis. The difference being that we'll finish off with haploid cells that are not identical to each other, but different because of that crossing over that we did during prophase 1. Now, as I mentioned, what is pretty much most important to the AP people is being able to compare the two. And most importantly, it has to do with, what do we see happening during meiosis 1, and then what are the results of meiosis versus mitosis. And again, I've got that extra handout posted on Schoology with the illustrations. The 3 major events that are unique to meiosis all happen during meiosis 1. And those include crossing over during prophase 1. It includes the fact that during metaphase 1, we have homologous pairings of chromosomes. So again, a little 2 by 2 lineup. And then the third thing is during anaphase 1, we see a separation of those homologous quote-unquote X's versus the sister chromatids breaking apart. Sister chromatids breaking apart would be like either half of the X, going in opposite direction. And we don't see that until, meiosis 2. All right, and so here's the illustration in the notes, but also on that extra handout that just does a really nice job, I think, of separating and showing you the differences between mitosis and meiosis. All right, now for the important part that in some ways is very new in terms of what you've heard before. Probably the most critical thing about meiosis is that it introduces genetic variation into our world. While mutations, which we'll talk about much more in the future coming up here, are really important, and these are natural. Mutations are not always bad, right? They happen naturally. These are ultimately the source of genetic variation, right? Mutations are the source of genetic diversity. It's why we all have different versions of genes known as alleles. I hope that word sounds familiar to you. But sexual reproduction and sort of like this reshuffling of alleles during sexual reproduction is what really makes us all so unique and results in every single member in a species being totally unique in terms of their genetic complement. So while we definitely need mutations to start out giving us some differences, it's meiosis and it's sexual reproduction that really makes us all different from each other. And what I'm getting at here, and we'll talk about at the end of these notes, is that this is the beginnings of talking about evolution. So sexual reproduction is going to really use three mechanisms to give you more genetic variation. The first one is called independent assortment of chromosomes. Now we want to be careful because next week we'll talk about Gregor Mendel's law of independent assortment, which is a totally different thing. So independent assortment of chromosomes means that when the homologous pairs line up at the metaphase plate, so during metaphase 1, and they line up in this little 2x2 pairing, all right, so here's a homologous pair, and then right beneath it, here's a homologous pair, and then beneath that, here's a homologous pair. You know, each of these pairs is one maternal and one paternal chromosome. But realize, and I drew it the way I did here on purpose, that there's not a defined way that they will line up. In other words, you don't get a boy line and a girl line. It doesn't work that way, right? Each of these pairs, these 23 pairs, technically lines up independently. It's this independent assortment idea, which means that any given time that you do meiosis, maybe you do get, this black one on the left and this blue one on the right. But at the next pairing, I've got blue on the left and black on the right. And then again, blue on the left and black on the right. But the next time your cell does meiosis, these might actually be lined up differently. And so at the end of meiosis 1, you have these two daughter cells. They're going to be a random mixture of maternal and paternal DNA just based on which side they randomly lined up on when they lined up during metaphase. So that's the first level of variation in meiosis, is that there's no defined way that these are going to line up during metaphase 1. And so there's no defined way or consistent way that they're going to be separated during anaphase 1. Now, as you can see in this diagram, if a cell has an N equal to 2, so 2 chromosomes, right? And then that would be a haploid version, so the diploid version would carry 4 chromosomes. Then we say that the possible combinations in independent assortment is 2 to the nth power. So in this case, 2 to the second power, or 4 different ways. And that would be, 2 possible arrangements for 2 chromosome pairs. And you get something like you see in this picture. But human cells have 23 pairs. So in humans, there are 2 raised to the 23rd power, or 8.4 million possibilities of outcomes that you can get just because of independent assortment of chromosomes. And that's just the first layer of genetic variation that comes into play during sexual reproduction. Second layer is the one you already know about, and that's genetic recombination. Right? Independent assortment gives us a collection of gametes that are new combinations of our parents' chromosomes based on how they lined up during metaphase. But also during prophase 1, don't forget that crossing over occurred. Crossing over occurred to generate these recombinant chromosomes that have never been seen before. Right? A new mixture of your parents' DNA that was not together before on a given chromosome. And contrary to what we teach freshman year, and I drew this earlier, crossing over doesn't always look just like that. Because what happens is crossing over can actually happen at any point along these chromosomes, and it will happen at multiple points along those chromosomes. So at the least, crossing over probably looks something like this. And it probably crosses over at even more positions. So you get a really big jumble or shuffle between the genes when crossing over occurs. And that's layered on top of the independent assortment idea that we just finished talking about. Now then you got a third step. And the third step is called random fertilization. Random fertilization is this idea that the pairing of a particular egg with a particular sperm is completely random, right? Um, uh, so I'm assuming you guys learned this in health class or something at some point. But an ovulation event, gives us one egg, and at a time, basically. But males will release millions of sperm. So which one of those sperm cells actually fertilizes the egg is completely random, and gives us lots of diversity. Now, if we put together numbers here, right? So again, each gamete is one of 8.4 million possibilities that we mentioned a couple slides back. So a fertilization event produces a zygote that is about one of 70 trillion possibilities, right? 2 to the 23rd times 2 to the 23rd. But we never even know which of these millions of sperm that's going to be, which means this number gets even bigger. And to really make your head spin, if you think about it, this is only, suggesting one male and one female. We also really don't know which male and which female in the human population are going to mate together, which is another layer of complexity that makes this number even bigger. And we don't even try to quantify what crossing over does to this 70 trillion possibility number. So essentially, every individual is unique because we have an uncountable number of possibilities that could give rise to you. And again, when we look at sexual reproduction, we talk about 3 layers: independent assortment of chromosomes, genetic recombination, and random fertilization that contribute to this uniqueness. So here's another picture of crossing over and what would happen in meiosis. Notice here they're showing a couple different crossing over events that gives you these totally unique chromosomes. Then they separate and you get these potential gametes that can then be used in cellular reproduction. So before we move on from this slide, let's remind you, there's a huge difference between the results of mitosis and meiosis. Meiosis is going to give us 4 daughters that are all haploid, and they are genetically unique. Compared to mitosis, which would only give you 2 daughters, which are diploid and are genetically identical. And finally, I guess it's going to be your job to watch a little bit of Elf. I did have a clip in here for us to watch. But the last thing to bring up, really for later more than anything, is that this is very significant in terms of evolution as well. As you might recall, when we talk natural selection, organisms with adaptations that better suit them or fit them to their environment are the ones that survive and reproduce. What we're ultimately saying there is the organisms whose genetic variation makes them a better fit to their environment will survive and reproduce. But don't forget that environments change, right? And so if an environment changes, what was once really well fit to that environment might not be well fit anymore. So what do we see if that happens? Well, we need genetic variation for species to survive, right? Because if the environment changes, we need unique individuals who might be better suited to that new environment to survive. I'll give you a really, really silly analogy, but I think a good way to think about this. I would say if you go to the pool, public pool in the summer, and there's this guy there who's just really hairy, right? He almost looks like a bear because he's got so much body hair. You're probably going to think that's kind of gross, right? And full-on body hair is maybe not the most attractive characteristic in a guy. But let's say that something happens and we enter another ice age, and all of a sudden that man is now better fit to that environment because of his body hair, his fur that he has. He stays warmer more easily, and he survives better, which means he can protect you better and your kids better. And so all of a sudden, what was not attractive, a full body carpet, might become attractive because Now it makes them better at surviving, right? And without genetic variation, you wouldn't have those guys who are different to begin with. So we need genetic variation, right? So that we can continue to evolve. So again, genetic variation allows evolution. So it becomes very critical, and that's not, a major point of emphasis for now, but obviously when we start talking more about evolution later, it will certainly matter a lot. All right, thanks for listening. Next class, we'll see if you guys have questions about this stuff, and then we'll continue to move on into genetic inheritance.